Welcome everybody to the March 4th working group meeting and mini seminars. We have two fine speakers lined up for today. I can't believe it's already March. It's a year since I last traveled somewhere. I'm sure other people are feeling the same thing. Here in Indiana, they've just lowered the vaccination age to 50. So I'm signed up to have my first shot next week. I know that varies a lot from place to place, and I'm sure you're having your own set of experiences on that. I'll remind you that the meeting is going to be recorded. It's live streamed, and it will be made publicly available. And so please don't say anything that you don't want to be out in the public. And let us know if there are any issues with that. As usual, the people on the organizational side are myself, James Glazier, Reinhard Labenbacher as co-leads, Jim Sluka, and Bruce Shapiro. I'm sure you've heard from all of us in various ways. And here's our contact information. And please let us know if there's anything we can do to make this meetings or the working group work better for you. You've heard me say this again and again. We have a Slack channel. IMAG MSM has a Slack channel. We'd love to have people participate in the Slack and help us make it work. There's also the IMAG MSM wiki that always needs love and editing. And so people's help with that would be much appreciated. Suggestions for additional content would also be much appreciated. I want to remind our speakers, who are very generous in coming to speak to us, that we only have on the order of 15 to 20 minutes for your talks. That's a very short amount of time. And I know it's difficult to say the things that you want to say in that period, but these are teasers. You're always welcome to come back and present again, either about the same topic or about different topics. The main goal of these meetings is to make connections, to stimulate conversations and ideas and collaborations. And I understand that it's hard to present all the technical information in such a short format. As usual, we really appreciate questions from people who haven't asked questions before. There's a lot of expertise in the room, and we'd love to hear from all of you. In the main session, we don't have time for many questions, again, because of the format. And so we appreciate that the technical questions be held for afterwards. Starting last week, we had produced these post-meeting breakout rooms for half an hour after the main meeting, which allow people to chat with the speakers in more detail and ask those technical questions which might not fit in the main meeting. And I will give a 5-minute warning to our speakers just as a reminder on the time. Our upcoming meetings, PBPK modeling in drug discovery and development, more clinical perspective. Veronica Zaritsina is going to give us the first of the breakout subgroup reports. Preliminary report on the subgroup plan for the innate and adaptive immune response. They've been making really impressive progress on that subgroup, and we're very pleased that she's willing to make a preliminary presentation of that work. March 18th, we have Yannis Kivrakidis, who of course is very well known for his work in data-based inference of mechanistic rules. And we have Ashley Ford, Recipt, who's going to be talking about multi-scale simulations of lung fibrosis. We are still looking for speakers for March 25th, and there's a slot open for April 1st. We have some people lined up. Coming up, Denise Kirschner has agreed to give us a talk, although the final schedule isn't fixed for that. And I'd love to hear your suggestions for future speakers. And again, suggestions outside of the mathematical biology community, experimentalists who are doing interesting work that should be modeled, people who are working on data sets, people working at other scales, clinicians who could tell us what they need from models. people in any of these areas would be most welcome. So without more ado, I'd like to turn over the meeting to our first mini seminar speaker, Jiang. I don't know if you are speaking jointly with your colleague or whether you're going to be giving the presentation, but please welcome our speaker, Jiang from Georgia State University.
Okay, thank you everyone. Let me see how I figure out how to share. I'm the one going to be talking, but Ling is really the one who did all the work. That's why her name's here too. So I'm going to talk about mucociliary mixing and transport, which according to James, that's not touched upon so far. So let me first talk about why, what's mucociliary clearance for those who are not very familiar with it and why we're interested in it. So here's the cartoon of our airway. When we breathe, particles go into our airway and eventually to the lung. Particles include aerosolized virus particles and bacteria and dust particles and allergens, right? And those particles come in and land on, float in the air and land on the mucus layer, the thin fluid, sticky fluid that coats the surface of the epithelium. The epithelium consists of the ciliated cells and goblet cells and some basal cells as well. The ciliated cells beat with the cilia protruding into the mucus and beat and try to move. It's like a sweep, sweeping the mucus layer and eventually push the particles, the foreign particles out from the larynx. Here's a microscopy view of a artificial tissue where you can see all the cilia beat by your eyes. And what we want to do is really want to understand where the particles go and how they move in the airway. And here's a closer look at the cilia. They are really these long, slender protrusions from the ciliated cell into the apical surface. And they consist of clusters of this, the clusters in the dimension of 8 to 10 microns in a patch, and typically 100 to 200 cilia together. And they are essentially microtubules. This ring of 9 microtubule doublets wrapped in membrane. And they are anchored at the base and wave around as the doublet microtubules slide back and forth against each other. That's how they generate the motion. And the cilia, when they move, they have an active stroke that's move one way and then come back the other way a little taller. So it's really like this kind of a 3-dimensional rotation, but it comes back a little taller. This kind of a cyclic beating, and with a frequency about 18 to 20 hertz, depending on the animal, depending on the disease conditions as well. So typical number that we use is 18 hertz for this beating. And this cilium have their tip stick into the mucus. They are submerged in the liquid, but there's the classical picture of that mucus is floating on top of this liquid, on top of the at the tip, near the tip of the cilium. But the more modern picture is that it's actually the fluid is rather complex, have many different kinds of mucus, many different there, and we are going to treat this as a homogeneous fluid. So the field has enjoyed a very long history of mathematical models, starting from the 1950s for flagellum or cilium, how different force can generate that kind of a beating shape. And here's a collection of recent work as a finite element model using the elastic network inside to model the dining and microtubule interaction. This is our work from a few years ago to simulate what factors of this beating process contribute most to effective transport. And here's a picture of a larger field of 400 cilium acting together. And when they move, when there are many of them, they don't always move in synchrony. So you see in this shape, it's actually, there's a metachronal wave as they move around. So those are all factors of consideration, but most of the mathematical models have been in a much smaller scale than the relevant tissue scale. Here's a recent paper studying human bronchial culture, and the goal was to compare between healthy and patients with asthma and COPD to see what factors are changing in their mucociliary clearance. And they used super fast microscopy to demonstrate that long and short time scale and long and short range patterns of cilia beating and mucus flow. And you can see in the centimeter scale, you see these large scale vortices. In the small, maybe 10, 20 micron, 100 micron scale, you see the small rotations. So when we saw this paper, during the COVID period, we had this observation of SARS-CoV-2 target ciliated cell, and some even suggest they target goblet cells, right? So we really want to understand how virus particles move in the airway. In the beginning of the infection, as the infection progresses, they kill the ciliated cell, the cell density change, the cilia density change, how that changes the particle movement. And if you also target the goblet cells, if you change the mucus, increasing the mucus, decrease the mucus, how that change the propagation of the particles. So our model is really to simplify the rod, the cilia into simple rods. And so it's a rod with different heights rotating. And in this projection on the top view, you can see that this is asymmetric, not just left and right, but also top and down. And we prescribe its motion. It's rotating at 18 Hertz, and the maximum length is 7 microns. And then the mucus is a simple fluid that follows Stokes flow. And the interaction is between mucus and cilia is through that force. Every particle, every position along that rod as they move, it drives it exerts a force on the fluid around it. And this is using a regularized Stokes method. So we apply no stick boundary condition on the bottom, all the cilium is anchored at its base and all the other boundaries are free. And we simulate a single cluster about 8 micron in size, about 9 coarse-grained cilium, and multiple clusters with varying spacing, and multiple clusters with varying different phase lags as in synchronized versus asynchronous beating. And we also simulate large tissue with varying cilia density. And here are the several model parameters. And in particular, the frequency is 18 Hertz. The length of the rod is 7 microns, as I've mentioned. So here's a quick picture when I have only one single cluster. And the top row is the top view when we look from top down. And the bottom row is the side view from the side. And we color the top virtual particles, several layers of virtual particles blue, and the bottom ones red. And there's actually on the very bottom, there are the black ones. And as we move in time, they beat in time, 5 revolutions later is 1.38 seconds later, you see this already some vortex forming. And the particles seem to bloom up a little vertically as well. And 400 revolutions later, 11 seconds later, you see this very large scale, rather large scale vortex, and with the top particles travel a lot further than the bottom particles. And really the black ones near the bottom, they hardly moved at all. So we actually see the overall, the particles moved in the lower left and lower bottom lower left direction. And then they also moved up a little direction, right? There's mixing and there's some transport. So in order to see how they move around, we use the mean square displacement just to remind everybody the mean square displacement is given the time interval, you average the distance squared. And if that's linear in time, the time interval, it's a normal diffusion. And if it bends up with the coefficient greater than 0, then it's super diffusion. That's advection and other drift associated with it. If it's less than 1, it's sub-diffusion. You can see it's a damped, jammed, or caged in motion. So we can see in our different layers, we just catch the average behavior for different. Z equals 1 is near the bottom, Z equals 3 is closer to the up, Z equals to 5 and Z equals to 7 is really closer to the tip. And we see all of them perform rather simple normal diffusion, except for the bottom ones that are almost stuck. So when we put these 3 clusters together and we color these virtual particles in different colors, on the top view, you can see the particles from the left being switched from the bottom right. So the whole thing is actually going in this counterclockwise cycle, right? Every single cilium is beating in counterclockwise. And then when they beat together, you see all these particles, the whole field is being stirred to generate these counterclockwise swirls. And in 11 seconds, you can see some green particles that's on the left, on the rightmost, now travel very far to the left, and the black particles on the left have traveled mostly along this direction, and some of them actually made all the way to the right, right? So there's a lot of mixing, and there's some segregation of, there's a color stripes, right? So it's some kind of a segregation of layers or of particle travel. And if you look at the side view, you can see lots of mixing and also some transport upwards. So as they stir around, the particles tend to be lifted up and then tend to be pushed in the negative X and negative Y direction. So that's sort of the observation when the clusters are closer together and all the cilia beat in synchrony. And when we push them far apart, now I have to change the view a little bit. The top view now in the left column and side view is on the right column. As a function of time, again, you can see this is the center of my cluster, and my cluster is about this size. And these are the particles in the neighborhood. And you can see effectively, they don't mix up that much. If they are far apart, so my own stirring doesn't affect the guys farther away. And same goes in the side view. The vertically, you don't have too much mixing. Horizontally, you don't have too much mixing, not too much transport. And if we plot everything, the center of mass in X, in Y, and in Z, for those particles as a function of their distancing between these clusters, we can see that if they are close together, they have the maximum transport in the Y direction and Z direction. So in Y and Z directions, they have sort of the further apart, the smaller the motion, the transport. But in X direction, that's actually the direction that we want the directed transport, the direction that's out of the airway, that you can see the smallest distancing does not provide the most directed transport. It's an intermediate. The distance equals to 5D versus 7D that actually provides higher, more displacement, center of mass movement. So that's interesting. So we proposed as a optimal distancing for effective transport. It's like the tandem of transfer. I have to move my particle this way, and for you to carry it over, and that's the relay game. There's an optimal distancing in between. And if the clusters don't beat in synchrony, this is the picture that we saw in synchrony. They have a lot of mixing, a lot of transport. And if we have the different phase lag between these different clusters, and you can see that they have less movement and they have more mixing. And in particular, in this case, that the movement, the transport from one cluster effectively cancel the other, that they end up with only mixing and no transport at all. So we can plot that, summarize the results, and effectively there's asynchronous beating, enhanced mixing, but reduced transport. And we can look at the mean square displacement as a function of these different spacing, and also as a function of different layers in our particle. And we see super diffusion in the top layers, and depending on their distance, actually different layers have more more advection, and that's spatially distributed movements. Another thing that we looked at was using replicate function to see clustering or dispersion from these particles, because we see some kind of color aggregates, and we wanted to see whether they, over a long time, they actually cluster or they disperse. And this is the interpretation of this replicate function. And we see when we have, it's very hard to read, but the reference line, the gray line, is our initial grid line. And in 3D, we'll see everything is below the reference line. So it suggests when the clusters are close together in time, there's an overall dispersion. In this intermediate spacing, it's overall clustering. And when they are very far apart, they don't have either dispersion or they don't have no redistribution of these particles. So this intermediate clustering, I don't know yet how to relate it to this intermediate spacing that's most effective transport, how clustering is connected to transport. So that's something we're still trying to figure out. And the last picture is with different density. This is the 20% density of these clusters at a large tissue scale. This is 100 by 100 microns. You can see a lot of effective transport versus at half of the density, very little transport. So this is a very preliminary and very rushed introduction on our simulation to study particle transport and mixing in mucociliary system. And we show there may be an optimal distance between the clusters for effective directed transport. Asynchrony beats the reduce transport, increase mixing. And all these may be potentially relevant to viral or bacterial infection, where we really want maximal directed transport to get them out of the airway as soon as possible, and minimal mixing so that they don't get stuck onto the cell surface. And then on the contrary, we want in maybe drug delivery, we want the minimum, we want them to stay in, we want minimum transport and maximum mixing so they can take effect. So thank you very much. I'll be happy to entertain questions later.
Okay, thank you very much. I guess since we have 1 second, does the transport depend on the particle size?
We don't have that yet. Our particle are virtual fluid particles.
So it's a passive tracer that you're using.
Yes.
Lovely, thank you. So we'll continue with Our second talk. Our second talk is from Kuhl, Stanford University, Data-Driven Modeling of COVID-19, Lessons Learned. Ready? I'll turn it over to you.
Thanks, James. So yeah, I'm scales way up from what you just presented. So I'm going to talk about population modeling of COVID-19. So I'm going to try to explain what we did to approach this and what we learned actually from doing this throughout the last year. So you're probably all familiar with the compartment models that people use to understand the spreading of epidemic diseases or outbreaks. And we use here the SCIR model, which is the model with 4 compartments, just to repeat that for everybody. So we have a susceptible, exposed, infected, and recovered group. And the population transitions between these 4 groups with 3 rate constants. So that's the contact period. That's how people come into contact with one another. The latent period. So that's for people that don't yet show symptoms and are infectious. And then the infectious period is when people can infect one another. And the ratio between the contact period and the latent period is what we all know as the reproduction number. I think by now everybody knows this because it's been in the news quite some. So the reproduction number above 1 is an outbreak of the disease, and below 1 is actually that the disease would die out. So we did this pretty early in the pandemic. So we started out in mid-March and looked at different countries. And this is what we did for Germany in June. And this was nice because by then it had gone through this entire first wave. And you can see the infectious group, which is the orange dots from the case data, is already back to pretty much 0. And so from that, we can then fit it to the real to our model data, and you can see it's a pretty nice fit. So the orange dots are fitted to, or the orange line is fitted to the orange dots, and from that we can gain these parameters. So we did that actually in mid-April for all countries in Europe, and I'll tell you just in a second why we did this. So you can see different behavior already here. So there's countries, remarkably, for instance, the last one is Sweden. Sweden never implemented any politics to restrict the spreading of the virus. So it's actually a pretty steep increase in Sweden of a number of cases, whereas, for instance, the first one is Austria, that had a pretty strict policy, and the curve already bends down at the same point in time. So this was neat, and we could then plot that across Europe, and we can plot different parameters. So for instance, here on the right, you see the basic reproduction number. So that's the number that I just introduced. There's also parameters here like the initial community spreading. That's something we don't see. We can just extract from the model and the data. So this is something that happened underneath the first surface before the first case was recorded. And then in the middle, you see how well it was contained. So that's the affected population with respect to the total population of a country. So this is quite interesting for us to study and learn. But then we thought, okay, we actually usually in our pre-COVID life solve partial differential equations in our lab. And so we thought, okay, maybe we can use that. It turns out that actually it's quite interesting to look at the spreading of COVID, and you can do that with discrete network models. And this is what this is showing. So we create a network where we mesh all the different countries, in this case, Europe, and we do this with a matrix that is actually the graph Laplacian, and the weights of the graph Laplacian are the passenger air travel between different places. And you can see here in this graph in dark red, that's Germany, Spain, France, and Italy. So that's the most traveled countries in the European Union. And then there's others that are traveled less. And the thick lines are the thick connections. So that's locations where people travel the most. So say from Spain to Germany to France. And that's the edges in our graph. So with this network, we can then solve this SAR model that I just introduced locally for every country, and we can see how populations mix. And this was interesting because when we did this, Europe was in this discussion of whether they should open their travel restrictions. And you probably know that Europe, the entire European Union, actually closed its borders on the 3rd week of March entirely for external travel. So all the colored countries here were cut out pretty much from the rest of the world. And so then this was when they were saying, okay, when should we reopen this? And that's when we did the simulation. So you can see on top a simulation with the travel restrictions in place. So that's what actually happened. And we simulated it with our model. And then underneath you would say, what we studied what would happen if there were no travel restrictions. And you can see there's a much larger red, so a larger infectious population, especially in Central Europe, Spain, France, Germany, the Netherlands. Whereas if with the travel restrictions in place, you can see everything is more blue and actually less spread. So this was interesting. Was actually, the study was in the news because it was just about when Europe was about to open. So this was got a lot of coverage in Europe. But this was also at a point in time when many models were criticized. And that was because they were actually just not suited for this kind of outbreak. And so what we decided to do is actually introduce something that would fix this error. So you probably remember that there was a lot of discussion about models, a lot of criticism, and all of a sudden, all the models were considered just useless. So this is when we thought, okay, why don't we introduce a dynamic contact behavior that depends on political measures? And that's what you can see here in the middle. We introduce a dynamic contact rate beta, and we make an ansatz that is a hyperbolic tangent function that you can see on the right in this plot. So it starts out with the basic reproduction number R0. It gradually transitions to the reproduction number in lockdown RT. And the time by which it transitions is this T star and T. So T star is when this happens in time, and T is how quickly this happens. And so we have 4 parameters that all have a meaning, and we can actually fit them and see how they relate to politics. To do this now, it's no longer easy to do by hand. And so we consult our friend Tom Bayes and the Bayes theorem that you can see here. So essentially what we do is we make prior assumptions for the parameters. So that's the turquoise color. And we assume a likelihood, so that's orange. And then the output is a posterior distribution of our parameters. This was when we submitted the paper on the effect of this tangent function in Europe. And you can see the same countries here. On top on red, you can see the reproduction number or the contact that starts high. So for instance, for Austria at 4, so that's a reproduction number of 4 at the early outbreak. And then it goes down to something below 1, and that makes this curve actually bend. And so we submitted this paper, and then we said, can we predict it? So all you see in gray here is a prediction, and we predicted 3 scenarios. The first one was leaving it closed as is. The second one, the light orange, is gradually opening, and then the orange, the dark orange, is opening immediately. And then while the paper was under review, actually we run the time went on, and we had these dots that we could then in retrospect retrace. So what you see on the gray on every graph is what happened after the submission of the paper that we had made these predictions. And you can see that most countries actually follow the behavior in complete lockdown. So most dots in the gray window actually lie on the prediction by just continuing lockdown. So then we thought, okay, what does this reflect? And we have this nice function. What can we do with it? So you probably know that air traffic dropped substantially, in fact, in Europe by 95% within only 10 days in the middle of March. And when you look at the air travel, and that's the dots that you can see on these plots, it takes the same kind of functional form as our reproduction number. So it has this tangent type function where it starts at 100%, goes down to 5%, and there's this transition. So they look actually quite similar. So again, the dots are the air traffic, the red line is the reproduction number, and the lag between the two we call the responsiveness. And that's something we learned from the data. We did this not only for air traffic, but also for cell phone data. So you probably remember there was this big publication or publicity of Apple. They recorded the mobility data of people during COVID. So we used those data, and that contains driving mobility, walking mobility, and transit mobility. And then we had the air traffic. And all of them follow this kind of tangent curve that you can see there. And on red is the fit of our reproduction number, and there's the responsiveness. If you do this for all countries in the European Union, you can see that the mean responsiveness is 17 days. So that's a little over 2 weeks. And you can also see that different countries responded differently. So if you look at the last column in this graph, you can see Sweden on the bottom, and I already mentioned Sweden. So they didn't really do a whole lot, and you can see their responsiveness is much, much slower. So it's about 24 days. And then there's other countries that responded only exactly 2 weeks and some much faster. So this is kind of interesting because it actually does relate to the politics in different countries. This was at the time when the first people talked about asymptomatic transmission, and it was actually back then pretty critical, and people didn't believe in it. And it was interesting because we teamed up with a group of people that are on this paper that's listed below. So that's Jay Bhattacharya, Aaron Ben David, and John Yadanis. And they're all at Stanford, and they did the first seroprevalence studies in Santa Clara County. And you can see that listed here as one of the 9 locations that we analyzed. And that was by mid-July. And what the study found is that there was an undercount on the order of 20 and above. So what the undercount means is that we count one person, and in reality, there's 20, where 19 are asymptomatic. And so that's quite a huge number. So we thought, okay, we can't really confirm this. This was all over the news because people were very critical about this number. And so we said, okay, can we at least visualize this? We can't see it, but can we visualize it computationally? And this is what we did. We added another box to our model. So this is this turquoise box there. And so what this does is from the exposed group, from the red, people can either become symptomatic or they become asymptomatic. And that's our only distinguishing parameter. So that's a fraction of symptomatic to asymptomatic. And that's essentially the undercount. And from that, both recover. So these 2 disease parts can have different disease dynamics. So it can be infectious for different times, for instance, because they're actually simply more outgoing, more infectious, they cough more. So there might be different dynamics individually, but we can actually derive all the equations analytically for these for this extra 5th group. We can also just fit it to the data. So this is for the Netherlands. And if you remember, it has an undercount of 5. So for every healthy person, they counted 5. For every symptomatic person, they counted 5 asymptomatic. You can fit the orange curve and we can predict what the asymptomatic population would look like and how the total recovery would look like. So in this case, of course, much more people would have recovered than we would know from the databases that we look at. So that's interesting. And you can do this for all places. So for instance, Santa Clara County, so there was the first study. New York did a study where they just studied people that walked into grocery stores, took their blood and did seroprevalence of them. So that was one of the first. And then there was also a famous one in Germany in Heinsberg. And so we compared all of them and we found they all look pretty similar. So all have kind of same dynamics. So the first one here is the reproduction number, the red plot. And then underneath you can see these different boxes color-coded by their box. And you can see that the asymptomatic and recovered group look pretty similar for all these groups. We can also actually, because we do Bayesian statistics, we can go back and say, hey, when did the first person show up with the disease that was not properly recorded? So in Santa Clara, we found that would be on January 20th. And we found that actually way before it was confirmed that there were cases before the case that was reported on February 23rd, I think. So this actually matched that quite well by doing Bayesian statistics. So then we were contacted by someone, and this was actually the general attorney for Newfoundland. And Newfoundland is a province in Canada. In fact, it's the smallest province. And you can see it here as the center of this network. Newfoundland is Labrador and Newfoundland, and Newfoundland itself is an island. And so the island had at some point in May, no cases of COVID. And so they had closed everything. So they had completely cut all the connections you see here to air travel. And so then their general attorney was actually taken to court because someone challenged their travel restriction. And so then they came to us and said, can you simulate what would happen if we reopen? So we did that. We had 3 different network models that you can see here. Red is for all of North America flying to Newfoundland. And then light blue is for only Canada. And for dark blue, it's only the Atlantic provinces close to Newfoundland. So that's only 4 provinces. Again, we create the graph Laplacian from air travel statistics and then simulate the outbreak in all of North America and the people that come into Newfoundland on a day. And this is the plot. So the color code here tells you how many people from that state would come to Newfoundland on a given day and be either exposed, so in the red bin, or infectious on the orange bin. So these are the 2 different plots. We can just add that up and we can count how many people would come to Newfoundland and bring the disease. So this is less than 1 per day. But if you open, on average, there would be some people within a couple of weeks and they would then induce the disease. So this is the same thing in just a plot and with statistics. So on the bottom, you see the different network models. So the North America, Canada, and the Atlantic provinces. And on the top, you see what would happen if people would perfectly quarantine by 50% and quarantine by 0%. And so we can quantify what would happen at which day if they would reopen the country. And what happened then is, this is Proton Rahman, one of our co-authors. He testified in court and we actually won the court case. So that was actually decided that it was the right decision to keep Newfoundland closed. So this was a very last real paper. And then by the end of the summer, when this court case was announced, we offered this class actually at Stanford. And the idea of this was that we were thinking, why can't our students calculate what would happen if we reopen campus? So you're probably all aware that there was big discussion in the fall, a lot of campuses keeping closed. In fact, Stanford is not yet open and has been closed for a full year to undergraduates. Many residential campuses have been very controversial in opening or had just closed right after again. So we said the students can calculate this themselves. We make a class and we give them a grade and they have to come up with a solution.
I'm sorry, 5 minutes?
So here's a group of our students. And so we challenged them to come up with a model. And they first looked at the data. So this is some of the results. You might recognize your institution here. If you don't, it's probably because they didn't have a high number of cases. If you do, and it has a high number of cases, don't worry, it actually most of them managed the disease quite well, which was not clear at the beginning of the fall quarter. So you can see Notre Dame has a high number, Wisconsin, UIUC, University of Florida, Clemson. So these are all campuses that have 4,000 cases or more. And this is the incidence. So the incidence, by now, I guess everybody knows what it means. So we plotted as a 7-day incidence. So we take the 7-day sum of cases and divide it by 100,000 people. And you can see that smaller campuses now, like Notre Dame, actually pop up because they have a higher incidence per student body. And then Wisconsin, Clemson, and Arizona had the peak cases. Here's a plot of the campuses that we looked at, a total of 30. And you can see in green, the case numbers increasing and then decreasing again. In red on top, you see the reproduction number, and in the bottom, you see a table. And I would like to direct you to the last 3 columns. So the third to last column is the recovered fraction, and that means how many students actually had the disease throughout the fall quarter. So say Clemson has 0.22, which means 1 out of 5 students had become infected throughout the fall quarter. And then the last 2 columns are the days to campus peak from campus reopening and the days to county peak from campus peak. And this is interesting because you can see them in this graph for 30 campuses. So the dashed line always represents the opening of the campus. The red number is the number of days until the campus saw a peak. And all of these are within 2 weeks. And then the blue number means that the campus peak leaked into the county. So actually that's kind of our definition of super spreading. It's not just creating a lot of cases, but it's also spreading into the county. And the red number also sees actually peaks within a week or 2 from campus peak. So this, as you can guess, got a lot of coverage because people were just interested in seeing whether campuses are super spreaders. So it was all over the news. And this was in a way just an analytical study to show, okay, within 2 weeks, the campus sees high numbers, can bring the numbers down. It leaks into the county, but the county for most places couldn't bring the numbers down as drastically as the campus itself. So it was kind of a kind of good and bad news. So with that, I just leave this up here. I don't think I need to summarize this. I'm happy to discuss things in the meeting. I think our main takeaway from what we've done is that we can model the dynamics with dynamic parameters, and we can model the control and correlate the control using actually what we've just shown and correlate that actually to political actions. And that can help obviously to select successful reopening strategies. And I'm happy to answer questions in the after session. These are the people who contributed it. Thank you.
Okay, thank you very much. Really appreciate that. We have time for 1 or 2 general questions to either of our speakers. People feel free to speak up. If anyone have a hand raised, I'm looking. Hold on. Jacob, please.
Okay, this is actually a very interesting one. You actually noticed that the numbers reported are incorrect. This is something which is interesting because this is something that needs attention, and you did pay attention to this. You said that each reported case is worth 5. This is where, is this the number you used? Did you try multiple of those values?
You mean the asymptomatic transmission?
You said that there was a publication that says that each reported case is worth 5, and then you incorporate that number, if I recall your talk correctly.
That's right. So for the Netherlands, so this is based on blood samples, right? So this is seroprevalence. So people did blood sample tests, how many people had already had the disease. And so the one that I showed was the Netherlands, where this is about 5 to 7. There were also other studies that reported much higher values. For instance, if you look at Brazil, had 5 to 27, or Idaho, 10 to 17 times. Those are numbers, of course, during the early pandemic. So I would think now these numbers go down because we test a lot more. So we identify asymptomatic cases. So for instance, for the campuses, most campuses test every student every week, as you know. And so there, these numbers I would expect they would be like on the order of 1 or maybe 1.5. Yeah, good question, actually.
There is a continuation, but we'll talk after.
Great, yeah.
Other questions? I had one question myself, because we, of course, like everyone else in the mathematical biology class, did this kind of modeling, not with the kind of effect that you did, which was that you show the active case numbers. But if you look at how the active cases are determined, they look at it at time of incidence, and then very often they simply assume that the case is cleared after 3 weeks. And so that the active case reported number is usually not a reliable number. Were you actually fitting active cases, or were you only fitting new cases or hospitalizations?
That's actually a very good point. Let me just share this again, and you can see exactly, for those of you not familiar with the problem. So problem is really that we try to fit the reported cases and the recovered cases. And if you try to fit the recovered cases as well, so that's the parameter C here, then actually you end up getting very long periods. And that's really a problem in recording. And that we try to first initially fit the data to the reported case. And we realize, man, this is like 3 weeks. So nobody is infectious really for 3 weeks. And we know this from the disease and from the way the disease is reported. This is really a reporting issue. And so then we went back and really from then on only used, here you can see that the blue dots are not really fit by these curves at all, right? And that's exactly the recovered. But then we started to just actually only use the infectious population, and that worked quite well, and use clinical values for the length of the disease. Good point, yeah.
Okay. Anyone else? Anyone from our silent audience have a question or two?
I do have one question if there is time.
Please.
Thanks to speakers. This is Asan Khwarizmi from Brown University. So I have a question for Ellen about the model that you're using. So I have seen so many people, so many different papers that they are using different models. So my question was how you trust the model that you're using, especially that you said that, for example, you started with the SEIR model and then you got a lot of criticism and then decided to add the asymptomatic into it. And I've seen so many different variation of the model. So, so which one is the best to use? So do you have, in the lessons that you have learned, can you share with us which model to pick? Thanks.
That's actually a good question. I see a lot of people nodding. So I think that's the golden question, actually. So what we've done is we have naively started, so I'm not an epidemiologist. So we've started this, like James said, with just very simple models, like we do in mass bio. And then we have gradually added things that didn't work. And I think that's in a way how modeling in general should be. I mean, you start simple and then you add things and add complexity. And then as you do that, you actually learn more about how the models work. And I wouldn't say there is the single one model that could do everything. There's another approach, of course, right? There's people who come with like a gazillion types of parameters, and they have huge models. You can model every population, every age group, right? People have different age groups. How do they interact? And everybody's like, oh, wouldn't it be nice if you could do this? Yeah, sure, it's easy to do, right? I mean, you can add more equations, you can add more parameters, but the problem is, how do you define them? And I think James alluded or asked that question initially, right? I think what we have now is a model where we know what the parameters do and how we can define them confidently from the data, but we can probably not model things that are really parameter driven, although it would be possible technically to do. But I think that's just the art of modeling. And I'm not claiming we know how to do it, but I think that's the golden question that everybody has to address.
All right, thank you very much.
Okay, I hope some of our audience will be able to stay on and have more intensive conversations with our speakers. Two very different and both very stimulating talks. Let me just come back to our slideshow. I wanted to come up with a few questions for the general audience, which are going to come up. So these are now more on the business side. We very much would like the subgroup leads to begin to present brief preliminary presentations for our Thursday meetings. We have some of those coming up on the schedule pretty soon. Please let us know, if you're a subgroup lead, when you would like to present, and we'll put you on the schedule. We feel that we could do better publicizing these Thursday mini seminars. And so ideas and help for publicizing these would be much appreciated. Media where we can get the word out about these meetings to make sure that our excellent speakers get the audiences they deserve would be appreciated. And as always, we would appreciate your suggestions for speakers. Thinking outside the box is most welcome. Experimentalists, clinicians, industry, government, defense, any of these areas, animal work, all would be appreciated. We want to reach beyond our basic mathematical and computational biology communities. We have our bi-weekly steering committee meetings. If there are agenda items or suggestions for organizing the steering committee better, we would appreciate those, especially Reinhardt, who's the one who has the job of steering the steering committee. There have been a number of requests for more explicit statements of goals and tasks from the working group leadership. And we are willing to provide top-down ideas, but we would very much appreciate bottom-up suggestions. Not really bottom-up, but more side-in suggestions, because it really isn't the top and the bottom. And so that would be helpful. Reinhardt and myself are always available to talk about these issues. Jim Sluka and Bruce also are available to talk about these issues as well. I think that is the main agenda. Again, if you are a new comer to the group and you haven't signed up for one of our subgroups, you are most welcome to do that. We would very much appreciate your help. Posting information, tools, ideas to the NIH, IMAG, MSM Wiki. Remember that if you have software and tools, you have to indicate that they're not endorsed by NIH. NIH provides the service of providing that site for us, but that's not the same as endorsing what we do. And those are things that we'd like to talk about. I wanted to maybe say a word or two. I attended one of the NSF prevent conferences / workshops last week. There were a number of very interesting speakers. They had speakers very much at the molecular level and many people doing population modeling of various kinds. Somewhat to my surprise, the only person who really spoke about the translation, the in-host infection translation from the level of molecule to organism was Denise Kirschner. And she's going to come and give us a talk very soon. However, there were excellent talks on some of the issues about the social aspects of modeling, how to organize models, how to build collaborative models from a variety of speakers. And I'm hoping that we could get some of those people to come and speak about the NSF Prevent initiative. There are going to be some relatively substantial RFAs coming out of that. And I encourage people to contact their organizers and make your views known about that. There may be, I don't know if there are any NSF Prevent people in the room at the moment who want to say anything about that initiative quickly. Is there anyone here who wants to make a comment on that? I found it a very stimulating meeting. But again, in-host was not their primary area of focus, more epidemiological. They were very interested in animal models. Okay. No. Reinhard, do you have any final business for today?
No, I don't. Other than encouraging everybody to sign up for subgroups, which is where a lot of the work happens. So other than that, I have no business.
So we have the breakout rooms open. People are free to move into them. So if Yi and Ellen are willing to move into the breakout room, then people who would like to speak to them are welcome. Certainly not required. But if you would like to have a chance to talk and ask some more detailed questions to Ellen and to Yi, they've graciously agreed to make themselves available for a little bit of time to be available. If you have any trouble reaching one of the breakout rooms that you want to get to, let me know, and we'll do our best to assign you to the breakout room and let you have your conversation. And with that, thank you. For the main meeting is over. The breakout rooms will stay open either for half an hour until people are done having their conversations.